researcher, soil and water researcher. It's quite similar to your work, but a bit different. My main focus is on soil and on the plant soil water ratios. First, I will tell you a few things regarding Shamir Research Institute. It's a research and development center located in Katzrin. As you can see, it has a variety of research topics and researchers, and this is part of the a national net of research and development centers all across Israel, especially in the periphery area. Concerning on how to improve the environment, the social affairs, and so on and so on. So it can be very diverse, as you can see. You might know two of these people. Professor Eyal Kurzwam is a researcher in environmental microbiology. Is focusing primarily on water treatment, but as you can see, is a PhD student, Dr. Ben Saar, which is already now a new member of our researcher guild. Some of you may know him. He's been around in your center, and he works on environmental microbiology. Besides water treatments, he really has a fond for ulva and other algae. So maybe we will focus on that in the future. I would like to show you another unit that we have under our institute, which is the Golan Agri Innovation Unit. This unit was it's operating since the last 3 years now, and it was cool to improve the work of our local in the Golan Heights farmers and growers, to help them get into much more profitable crops, and to improve their techniques regarding soil and water, which is mainly irrigation and fertilization. This is my department. We also have for plant disease, pesticides, and everything. So we try to help them in every kind of way. We also this unit depends on the triangle between a researcher, a local guide that works on the field, works with the farmers, and the farmers themselves. We all together work to get the simplest solutions for their challenges, and by during that we get new ideas for research and for improve our work as well. Let me introduce you to my favorite playground. This is the Golan Height. As you can see, the lowest parts of it are near the lake Sea of Galilee, Lake Kinneret, depends on where you're from. And you can get it from minus 200 meters below the sea level. And in the Hermon area, it's around 1,200 above sea level. I live just next to this 900 in Meron Golan. So we have a big difference in elevation, which also comes to it meets also differences in rain precipitation, in temperature in temperatures all the year around, and so on and so on. Therefore, we have a nice place to demonstrate differences between with the same crops all along this the plateau, so-called plateau. As part of our services for the farmers, we conduct a soil surveys before they plant. This can be very crucial in farming. You see, when you don't have a clue, an idea regarding what happens beneath your soil, beneath your your ground, you can have differences in water containing, in organic matter containing. You could have floods. You could have a lot of problems regarding infiltrating water differently. And farmers usually try to work in all their field homogeneically, uh, which also helps them to work in order to be economically reasonable. So we survey their plots before they plant the trees. We map using GIS, of course, to make sure that we have uh, all the significant differences between areas of the plot, and then we supply them with idea of how to plot which crops could be beneficial there or not. So it's a tricky business, but it's a very nice way to help around. And I'll tell you that it's very nice to just drive by all around this area and look at the orchards and the fields that are planted according to my advice, and to see them bloom. So I hope that each of you will feel this way regarding their work. By the way, if there are any questions, please do stop me. We have a lot of projects regarding precision fertilization. This is all part of the scope of precision agriculture. Since we know that we have differences inside the plot, we have differences between trees, varieties, all sorts of things. We need to irrigate and fertilize according so. And therefore, there's a lot of efforts today in order to first use remote sensing or other devices and sensors to measure the differences and to irrigate and fertilize according to it. It's we have a lot of some of the projects I will describe later in this lesson seminar. Okay, we talked about the soil itself. Now we need to realize what is the quality of our irrigation water. If it's saline, if it contains sulfur, if it contains a lot of organic matter, nitrogen, phosphorus, potassium. If it does, we need to irrigate accordingly, and we need to make sure that we have a time lapse of irrigation long enough between irrigation periods, so the soil itself could breathe, could have some time to adjust to this water apply. You will see some of these projects also later on. Soil erosion. Have any one of you ever confronted this situation? Did anyone ever notice something like that? That you can see how during a harsh period of rain, some of the soil in orchards, in fields, just next to roads, just flows with the flood. And you can see how there's a decrease in the soil in the field. So you will have less soil from year to year, and it's a very harsh problem, especially in heavy rain places where you have a big elevation problems. And some of our work is part of concerning soil health and we want to mitigate this problem. So this is part of our motivation to know how to irrigate right and how to grow our orchards right. Besides that, we have a lot of projects regarding soilless agriculture, from hydroponics to aquaponics, as you can see with the fish. I'm guessing that you guys have more experience with aquaponics than us. We work a lot with hydroponics and soilless cultures in net houses, in greenhouses, and so on. And we have a nice, we have a nice farm, research farm in Avnei Eitan, which is on the south of Golan, when we are now establishing different techniques of hydroponics systems. If we will talk later on in a few years, we can describe what the differences, the advantages, and disadvantages of each system. Okay, I tried to set this seminar chronologically. So I started from my thesis for my master degree. We were measuring olive leaf minerals, phosphorus and potassium. We measured the concentrations according to their visible and near infrared spectral reflectance. As you can see from the devices in the picture, we measured with shelf devices, on-shelf devices that measure reflectance from the leaves themselves. We have 1, 2, 3 different devices for different spectra. As you can see, on the bottom of the picture, you can see the olive leaf samples themselves after grounding and heating. And we try to, as I said earlier, we want to make efficient fertilization in olive. And therefore, we need to make sure that the measurement will be in situ, on site, and as much as possible leaf samples that we can take. The usual way is of olive leaf samples measured today is in chemical analysis in the lab, which it's time consuming, it's labor consuming, and therefore it's been used less and less than we desire, if we want to make sure that we have the precise fertilizing management. And we tested olive leaf samples, Barnea, that were taken from orchard from in fertilization experiment in the Gilat farm, which Gilat, as I will describe later on, is a farm of the Ministry of Agriculture concerning the agriculture on top and next to the desert. It's just next to the desert area, and they researched this subject. Over there, they had an experiment of olive trees grown in different barrels filled with substrate, and this is how they can manage their nitrogen, phosphorus, and potassium levels. We measured them in 400 to 50 till 1,000 nanometers, and from 1,000 and 100 nanometers till 1,700 nanometers, which means in the shortwave infrared. As you can see, we had a lot of, we correlated the same leaves. We measured them accordingly with the traditional chemical lab measurements. And we got pretty good results for, we had a cross-validation for nitrogen for was around 90% correlation, so, which is quite good, I would say. And for potassium, it was less, we had a less advantage here. And also phosphorus wasn't such a success. Well, it is a result. But we did show that after we dehydrated the leaf, the model performed significantly better. And also we did show, according to the work, that this Wisner spectroscopy can be used for decision-making in fertilizing procedure. Hopefully that it will get into different crops as well. And by the way, olive leaves are a hard study case because their structure is quite like a roof tile and very hard to get through. This is why we had to dehydrate them and to grind them. At least we had a protocol that suggested a laboratory spectral measurements, and this is what we did. Going further to my PhD work with my supervisor, Dr. Asher Bartal from the Volcani Research Center, which is the Ministry of Agriculture service for research. We measure the long-term effects of compost on the dynamic of both the soil organic carbon and the nitrogen pools. We all have, when using compost today, we all have the notion that it helps physical amendments of the soil. It helps both keeping the soil alive and refresh and keeping soil microbial organisms just in a much better state than they were. But regarding the nutrition addition of compost, it's still debatable. It's still on debate, and this is one part that we wanted to make sure to research. So we do know that yeah, getting into a much more sustainable agriculture production, we have to have a lot soil organic matter content higher and stable. We do know that soil that do have depletion of soil organic matter are under conventional management, and it's a well-known phenomena all around the world. And there is the notion that organic management tends to counteract the depletion of soil organic matter, and especially in arid soils. This is why we tested it both in northern Israel and south of Israel. The general objective was to investigate the effect of organic management. If you can see in this illustration, when using synthetic fertilizers, we only feed the plants themselves. We do it quite precisely, but we feed only the plant themselves. We overlook all the organic matter pool, the soil nutrients, and the microorganisms. All of them are overlooked. When we are using organic fertilizers, we feed the soil itself. We feed all those pools with the plant themselves. It's much more tricky. There are much more variables, but we know today that we can't leave out all this soil health. So we went to characterizing the influence of organic application management at different compost loads, and we measure it comparing it with chemical fertilizer, which we tested it for long-term effects on soil fertility and soil carbon and nitrogen reservoirs. On the short-term effects, we tested soil microbial carbon and nitrogen and other liable pools of carbon and nitrogen in the soil. So as I said, we tested it both in Neve Yaar up north, where they have a better precipitation, and in Gilat site, which it's on the verge of a desert, and they are under a much more stress of losing soil organic matter. There are differences also in, as you can see, differences in regarding to sand percent, the sand, silt, and clay percentage. Therefore, those type of soils are quite different. Regarding the clay, there wasn't a big difference between them, between 15 and 18 percentage. There's a vast difference in the rainfall between the sites in Gilat and Neve Yaar. As you can see, 220 millimeters per year are it's quite low, and it's on the verge of calling it a desert. Regarding the organic carbon content, we have a difference, as you can see, almost twice in Neve Yaar next to Gilat, and also the nitrogen, and so on and so on. We measured along compost doses. We measured in different compost doses. 0, which has no compost dose at all. It has a chemical chemical nitrogen addition, and that's it. 20, 40, and 60 cubic meter per hectare per year compost dose that were added to the soil. Each year for 7 years, it was measured, and we evaluated this application influence on the total organic carbon, as you can see on the top here, on the top. You guys can see my mouse movement. Thank you. And as you can see, there was an increase with an increase in total organic carbon and in total nitrogen in the soil compared to the compost dose. It was a total in total measurement. So therefore, you can see there's this increase in soil carbon and nitrogen pools according to compost application, which is quite crucial to continue with our work. We had an idea to measure whether the ratios carbon to nitrogen, which is very influential on which organisms live there, and which nitrogen processes are being are ruling in this area, in the soil. And we saw that as long as there is the compost dose increase, with compost dose increase, you can see a decrease in carbon to nitrogen ratio, which means you have much more nitrogen compared to carbon. Therefore, there's the enrichment in nitrogen is very crucial here. And prefer conservation of nitrogen over carbon. See? Okay. Then we had this soil fertility bioassay. This is called Gilat test. It's a test that by taking the sods and growing a plant on it. Here, the best plants, one of the best plants for this kind of bioassay is corn, because corn is actually a pump for water and nutrients. It uses whatever you can give it to him. You can give to him, and it influences its growth. And we measure the corn biomass in dry weight, in percentage of control. We took the control of with no compost application, and we measured all the plants in compared to this control. And as you can see, during we had this test at the end of each year of cropping. So we did see that almost in most of the years of growing, all the compost treatments had a much more corn biomass, around 100% more of the control of the non-compost applicated soil. And we did a multi-annual corn biomass after 7 years of growing, dry weight of the percentage of the control. And as you can see, um, with increasing of the compost dose, you have an increase in the biomass, in the corn biomass measure. And with a big significant, it's a significant and big correlation factor. And this is quite a notion, because a lot of people like to say that compost application does not improve the biomass of the crops. So here we can show that. That was in the long range. Now, we were talking about short range as well, that before compost application, and 1 week, 4 weeks, almost. I spent a lot of times in soil sampling in those dates, and we measured a lot of compounds of the carbon and nitrogen pools in the soil. And we can see this could show us a dynamics of the soil carbon and nitrogen. Regarding the total soluble inorganic nitrogen, that's the top graph, and the second graph we have a microbial nitrogen in the soil, which was measured in the microbial constitute was measured with the fumigation extraction techniques. And as you can see, along the time, you have big fluctuations in both those pools of nitrogen. That could be explained either by the compost application, by rain, precipitation, and sowing, and so on and so on. But you can see that the black markers refers to the non-compost application soil, and you can see it was most of the time. It was most of the time beneath in the both attributes of microbial nitrogen and inorganic nitrogen. But as you can see, those increases, you have a decrease in total soluble inorganic nitrogen in all of the soils with compost, mostly in them with compost. And at the same time, you had an increase, and significant increase, in microbial nitrogen in the soil, which tells us that there was microbial nitrogen immobilization, and it's a process that we consider that nitrogen was saved inside the soil in this pool, which is later on being used by plants. When we characterize the water extractable organic carbon, on top you have the cold water extraction. On the lower you have a sequential hot water extraction in 80 degrees. The samples were left in 80 degrees for a few hours. And this is considered to be a much more resilient carbon pool, which is have a tendency to be a slower release of carbon, but it stays for longer in the soil. So in here on the top, you can see the seasonal dynamic, but on the hot water organic carbon extraction, you have a significant fraction of the organic matter. As you can see by the measurement, it's 760 and 550 milligram per kilogram. Well, in the cold water extraction, we can extract until 200 at most. So the hot water extraction is a significant fraction. It has weak seasonal dynamics. It's much more stable, but it is sensitive in the long run for application of compost. In the long run, we do see differences between the treatments, and uh, with increasing loads of compost, we have an increased values of hot water extraction carbon. So this pool does increase with compost application. As we had a lot of conclusion. On the same site, we try to characterize the organic matter in the soil. So we use the excitation emission matrices of fluorescence and UV absorption methods to characterize the changes in soil labeled organic pools accordingly with the compost application. We tested the EEMs of fluorescence and UV spectroscopy, and we did it with a range of compost rates. We tested them using a parallel factor analysis for identify the chemicals fluorescent components of the soil organic matter under a wide range of compost rates. I do remind you, we are looking for a way to characterize whether our soil is at a good health or situation, and to measure the soil organic pools of carbon and nitrogen, both. We did it on the same soil samples that you saw earlier, and we monitor the measurements of both total organic carbon, total nitrogen, and dissolved organic carbon, which is all as mentioned as before. But we did measure both UV absorption at 250 nanometers and the fluorescence. The absorption of 254 nanometers is measured as a UV measurement of organic carbon in a cyclic formation. And we measured it both in cold water extraction and hot water extraction below. As you can see, we can see here the effect of compost dose on the seasonal dynamic in the upper one, which is the cold water UV measurement, and in the hot water where we can see much more stable measurement, component the array application, and so on and so on. We tried we identified 3 different components in the Parafac model, which describe the EM of the fluorescent of the soil extract, and we did use in the data set both cold water and hot water, and these three components described the changes of the carbon pool along time, along the procedure. We did using the absorbance at 250 nanometers. We found it is a very sensitive index for compost dose, which in every compost application it altered the absorbance at 254 significantly. Regarding the fluorescence intensity of proteinaceous components, which was proportional to their concentration in the extracts, it decreased with the increase of compost dose. With increase of compost dose, there was a decrease in those elements, and we found a ratio between between fluorescence and absorbance ratio which was the best parameter to test the influence of compost load on the composition of water extracted carbon, or organic carbon, obtained by both cold and hot water extract. Going to an ongoing experiment, which today I was there at the field. We looked the mango growers wanted to check how to fertilize with nitrogen with sense. Up to now, farmers have no idea. Mango growers in Israel have no idea how much nitrogen. When you ask how much nitrogen they need to apply. The doses went from kilograms per hectare till 20 something, 25. That were the higher. So there's a big debate on regarding how much we need. So we have a nitrogen application. We are continuing the work in lysimeters. You all know what lysimeters are, right? Or should I explain? No? Okay. When you measure properly how much water or fertilizer a plant needs, the best way to do it is to grow it in a non-soil without soil in a container using perlite or a different medium that doesn't conserve both the water and the fertilizers. And you measure both how much water and fertilizers you put in to this vessel and how much water and fertilizers are going out. And for that, you need to have a lot of piping and measuring all the water getting out of the pot. It'd be a very big pot. When you grow mango, it should be 50 liters at least, or 100 liters, depends on your experiment. And this is the proper way to measure how much water does the tree consumes. How much does it need? And you can test both how it affects the productivity of the tree, how much it affects the tree water consumption and water use efficiency, and so on and so on. So this work, it's a continuation of a pot experiment, which was made in Tzemach next to the Sea of Galil. And now it's been done in an orchard in the western part of the Sea of Galil. And it's on a Shelly variety mango in orchard. We measured them 7 treatments of non-nitrogen at all, 50 kilograms per hectare per year, 120 kilograms per hectare, 200. And we have 2 treatments where we're getting the nitrogen, the application, either with ammonium formation or nitrate formation. All other treatments get 50%, 50% ammonium and nitrogen. And the 7th treatment, which is quite special, most mango growers believe or they think that applicating nitrogen 1 month before the fruit picking can decrease the color of mango. You see, one of the major attributes of mango, as you can see, is buyers yourself, I guess. When you go to buy mangoes, usually you will pick it according to the color of it, which is not all the time true. In some varieties, green mango could be very delicious and great, but the public wants, and that's what the public will get. And so we tested whether this story is true, whether if we don't stop to we don't stop the fertigation of nitrogen before the fruit picking, whether it will harm the color. So we have 7 treatments, and as you can see, this is how the plot looks, the plot scheme. We have 7 treatments, we have 5 repeats, and we do it where since it's nitrogen is fluid in the nitrogen can move around in the soil. So we have a boundary roots. Each each block, sorry, each block is a tree, and as you can see, we have a lot of trees in there. With all the we have 20 trees per acre there. It's quite an experiment. Very heavily, we conduct there. We conduct this experiment for the third year already, and it's one of our major efforts. For this year, we measured both average weight of how much time do we have? You have okay, never mind. And I would say that we measure the nitrogen effects on both the average weight per tree this year. As you can see, there's an increase with compost application. Sorry, on the right, you can see according to treatments, and you can see that from some reason, the no nitrogen at all treatment has a higher fruit weight per tree. Let me just say that this year we had a really special low fruit weight fruit at all yield in these trees. It was all around the area. Hopefully that this year will be better. We also measure it according to nitrogen concentration in different issues in the seed. And I don't want to make you too tired, but I will show it there. I will run through it. We also measured the phosphorus percentage and the potassium. We also measured the how did the nitrogen application affected both side decay of the food, and we saw differences between the treatments. This experiment is still on the go. Hopefully, we'll get a result. We have another big experiment improving calcium and the quality calcium absorption and the quality of mango fruit with precision coating with polyphosphate. We apply the phosphorus in a phosphate form. This experiment is run by Ran Erel and Noam Alkan from Volcani Institute. Let me tell you something about the calcium transport to the fruits. Calcium is one of the elements which is heavily used by fruits, vegetables, and us. What does calcium does in our body? Does anyone know? Calcium is heavily used, contained in our bone, in our bone. The same with fruits. It's a main fraction of their stability of their outer membrane and the cuticulars. Sorry, it's a part of their so-called skeleton. And the lack of calcium can be spotted in many cultivars, from vegetables and tomatoes as well, in leaves, and also in fruits. When you have when you don't have sufficient calcium, you will see in apples bitter pit. In mango, you will see the jelly seed, as you can see here in here, and it affects shelf life, and of course, those mango fruits will be tossed and lost. When we're talking about orthophosphate, which is the main form of phosphate applied to trees and orchards, this is one. And when we're talking about phosphate, tripolyphosphate, longer cyclic, you have so many. Sorry, you have so many forms of polyphosphate extinguisher to be motor engine liquids, and so on and so on. We're using a polyphosphate applied from chemicals to Israel, KIL, and it's been used in all those crops that you see here: mango, avocado, cabbage, hydroponic, celery, lettuce, and even apples in Iran next to us. All of those experiments are run by Ran Harel. Is polyphosphate application, and it's checking how it changes the crops yields and quality. So we were looking into the mango responsive polyphosphate in the field next to Tel Katzir, on the southern part of the Sea of Galil. And we have both orthophosphate and polyphosphate. But we had a problem there. At first, we saw that there was a major improvement in color of the fruits, but near the end of the season, we had all the trees were sick and had pesticides. They were very vulnerable, and it appears to be that this plot was very sick, and had to be treated and not to be as part of our experiment. As you can see, having a field experiment is not easy. Something that you have to exercise and to improve. So we moved to Moran Lotem orchard, and we conducted since last year different experiment regarding polyphosphate. We had 2 treatments of polyphosphate. That the formulas are still not available to the public, so we will call them poly A and poly B. Orthophosphate and managerial area, which we didn't treat any of it. We didn't. As you can see, we have 5 repeats for each treatment, and we measured in the on the. That's another thing that I want to show you here on this slide, is the differences between test trees and the measurement of the whole plot. On the left side, you have the fruit weight sample per tree in kilogram, and in the warehouse, not the warehouse, the local factory that conserve those film, which means all the fruits, total fruits of all the plot we're taking into of the plot and the treatment, we're taking into account there. So you will have the measurement will be much more precise. On the right side, these are the two test trees in every plot that we have. As you can see, there's a bit difference between them. And that's one thing that whenever you have a field experiment or an experiment at all, you need to make sure you have either you have the best test trees, or you will have to measure the whole fruit in the plot. We measure the nitrogen in the fruit pulp, and we saw differences between them. As you can see, there wasn't any major differences in the average fruit weight, but we did saw that the poly A and poly B decreased significantly. The jelly
Nativ,
seed, yeah.
I'm sorry, but we ran out of time for the seminar. Can you just sum it in one, like,
Okay.
finish the sentences? Thank you.
Okay. I was trying to get you into my world in short while, but as you can see, we have a lot of work here regarding soil and the irrigation and fertilization. I know the connection to your institute is a bit slow. So I try to show you something a bit different than you usually work on. But I think we're all part of the same system when we act local and we need to think global. And this is what I was trying to show during this seminar. Thank you very much for listening. Any questions?
For question. We will take your contact from Yoav, and if some of the students have questions for you, they will send it personally. We would like to thank you for this seminar. Thank you very much. It was a brand new
Thank you very much.
for our students. Thank you.